All right, so what we're going to set up today, okay, we're going to set up our procedure and our lab and our essentially experiment that's going to run over the course of the weekend. Okay, what we want to do in this lab is confirm the force that is being used to draw water up a plant. Okay, we want to confirm what that force is. Now we know that that force should be what? No, I was thinking really one word. Osmosis. Okay, we know that osmosis in combination with the polar nature of water, but osmosis is what draws it up. The polar nature of water is what keeps it from falling back down. Okay, so osmosis is going to create this pulling force that's going to draw water from the roots to the leaves of this plant. Okay, what we want to do is confirm that that's the only force that's going to do it. Where is it that some people sometimes think other forces may be located? We talked about it the day we talked about. Right, that there's this root pressure and things like that that comes from the roots. Well, what we're going to do is we're going to eliminate that by cutting them off. Okay? So essentially what we're going to do is we're going to manipulate the plant's structure a little bit, okay, so that only the leaves can be producing a force that moves water. Okay? If we eliminate the roots, we eliminate them as a source of pressure that can push water up the plant. If water still moves up the plant, then we know the force that's doing that has to be located in the leaves. Everybody follow my logic there? Okay, so we will cut off a few leaves off of these plants, okay, making sure that we get some good stem left on there so that we can get them down into our water supply and they don't run out of water before Monday comes. Okay, we'll get those, and what we'll do is we'll put them in dyed water. We're just going to use food coloring. Okay, you might have done this like with a stick of celery back in elementary school. Okay, yeah. All right, so what we're going to do is we're just going to use these plants. They got some nice white parts, so the dye shows up pretty good in them. So we'll have that running over the course of the weekend. But there's a few things that we have to control. Okay, we have to make sure that essentially the conditions are the same for all the plants. So all the plants are going to sit in my window. Okay, so they're all going to get, hopefully decent sunshine over the weekend. Okay, they're all going to be the same temperature, all that kind of stuff. The big thing, though, is that we want to make sure that the water can't escape from our setup any way other than through the leaves. Now, that's going to be a little bit tricky. I can't just stick these things in a cup and leave the cup open on the windowsill because water will just evaporate out of the cup. Okay, I won't know if the water that disappears on Monday. So when I come back on Monday and I know, hey, there's less water in the tube. Okay, I'm not going to know whether that went into the plant or whether it just evaporated. Okay, so I've got to make sure that I've got a way to seal this thing up. So part of our apparatus is that you're going to go into the lab and you're going to, because I'm recording this and I can't have their face on YouTube. So I have to block the face up. Okay, so what we're going to do is we're going to make some dyed water in one of the 100 milliliter graduated cylinders. Okay, so you're going to take a 100 ml graduated cylinder and fill it to the 100 mark. Okay, then you're going to add, let's say, 3 or 4 drops of food coloring. No more than that. You add more than that and it actually could start to thicken the mixture. Okay, so we want to have 3 or 4 drops of food coloring. Your choices for food coloring colors will be blue and red. I used to put green out as a joke, but too many people used it. Yes. It was only funny the first few times, and after that it was just sad. Okay? Because on Monday they're like, Mr. Coder, some of our water's gone, but we can't see the dye anywhere. Really? That's so strange. Anyway, okay, so yeah, we're going to use these. We're going to use blue and red. If you want to mix blue and red, you'll have a nice purple. Okay, that's up to you. So we're going to mix 100 mils of that, and then, this is the key part, we're going to pour that into a smaller graduated cylinder. Okay. The reason we have to do that is our small graduated cylinder, well, all of the graduated cylinders, only have graduations up to about here. And we need to fill them pretty much to the top. So we can't measure how much water is above that if the marks don't go higher than that. Okay? So we're going to have a 25 mil graduated cylinder that we're going to fill from our 100 mil. Everyone okay with that? So we'll fill it up. to just about the top. Not quite the top, because you still have to stick the plant in there. Okay? If you fill it more than that, then water will go over the top, and you won't know how much you lose. Okay? So we'll fill it close to the top, and then what we'll do is we'll look at our bigger cylinder, okay, and we'll look at how much is left in there. I started with 100, and if I subtract what's left, I'll know how much is in here. That'll be our starting volume. That will be an important number for you to record. Okay, you have to make sure that you record your starting volume. On Monday, you're going to take your graduated cylinder and pour all the water back into the big graduated cylinder so that you know what your final volume is. And that way, we'll be able to calculate how much water was moved by the plant. Everyone all right with that? Okay. In order to create the airtight seal, or at least close to airtight, so that we're not losing a lot of water to just evaporation into the atmosphere, I'm going to give you the finger off of a rubber glove. I love doing that. That's like my favorite joke of the whole year. Okay, and you're just going to put it over the top of the graduated cylinder. Okay, so you'll just grab it and kind of just pull it down over the top. It creates a pretty good seal because they're about the right size for the 25 mil graduated cylinders. And you're just going to poke a hole in the top of it with a pencil. Okay, then you'll go and cut a piece of the plant off and stick it through the hole and as far in as you can, so that it's as deep into the water supply as possible. Okay, if you only stick it into the water this far, it's possible that it'll draw all that water out and then not be in the water anymore. Okay, and then our experiment kind of breaks down. So you need to make sure when you cut your plant that you get a fair amount of stem along with the leaves. Other things you have to be mindful of when you are cutting the leaves, okay, and that is that the leaves have to be cut underwater. So I'm going to fill up the sink with water over there, and I'm going to throw the plants in the water. You need to submerge the plant, cut it, and then put your thumb over the cut end before you carry it back to your table. Because if you don't, air will get into the xylem, and that will disrupt the hydrogen bonds of the water that is already in there, and it'll interfere with the transport process. Okay, so we want to put our thumb over the end so that it's kind of sealed. So you cut it underwater, put your thumb over the end under the water, carry it to your table, have your apparatus all ready, take your thumb off and put it in. Okay, do it really quick so that we don't lose or we don't get any air up into the xylem. Okay, most of the time it isn't an issue. Okay, but I've had people that have just carried it over with forgetting to put their thumb in. Okay, and then their experiment doesn't work. Right, so make sure your thumb is over the end. Put it in there. Okay, so this is about the size of the piece that you want to have. Get a few leaves, some big ones, some small ones if you can. Okay, but have a fair amount of stem below. Okay, you can see she's kind of got about 3 fingers width worth of stem there. Okay, that's about how much you want, and put that in, and then you're good. Okay, the final apparatus should look like that. Okay, a couple of leaves, 2 or 3. Okay, in there you can see the finger of the rubber glove here over the graduated cylinder. You can see the water's almost right to the top. Okay, and you'll put a piece of masking tape with your names on it on the graduated cylinder so you remember which one's yours on Monday. Okay, that's what your final apparatus will look like. Now, the other thing, once you have the apparatus built like this, then you're going to take out your phone and you're going to take some pictures of each leaf, top and bottom, so that you have some comparisons on Monday. Because on Monday, hopefully the leaves are going to look different. Okay? The idea here is that when we come back on Monday, they're going to be a different color, or at least parts of them. There'll be some evidence that the water has moved into the plant. Okay? We want to be able to compare there. Everyone follow? Okay. No, they won't live after. Okay? But they'll do the job for what we need them to do. Okay? All right. So we've kind of talked a little bit about our variables. Okay? We talked about some of the controlled. We talked about the manipulated. Okay? Responding variable, there's 2. There's 2 responding variables here. There's 2 things we're watching for. Okay? We talked about volume, and we talked about taking a picture of the plant for some reason. You guys got to figure those ones out. Okay, so for our hypothesis, okay, the if part should probably talk about how we think plants transport water. If they transport water in this way and describe what we're doing here, I would make sure in the and part you mention that we've removed the roots. Okay, that's an important part of the experiment. Okay, The then part should tell me, what are you going to see on Monday in terms of both the volume of water in the cylinder and the appearance of the plant? Okay, you got 3 analysis questions on this one. Okay, the first one is essentially just doing the calculation of how much water was moved by the plant. So your starting volume minus your initial volume. Okay, you can talk to some other groups, okay, and see if there why or if there, are any differences. Number 2, okay, what evidence do you have that water followed the path discussed in class? So that diagram we went over the other day about where water goes, okay, what things it goes through, okay, how it gets out of the leaves. What evidence do you have in the appearance of the plant that that's what happened? So kind of look at where the dye appeared in your leaves and talk about that, okay? And number 3, that's something we actually did talk about a little bit in class, okay? If we did this experiment but put salt in the water, how would it change the results and why? Okay, what would be different about what happened and why would that happen? Okay, that's what question number 3 is asked about. Then conclusion evaluation, same as always. Okay, questions there before we go across the hall? All right, we should be able to do this in, let's say, like groups of about 3 or so. I mean, I got 3 plants here, so we should have enough leaves for groups of that size. The graduated cylinders are in the back. In those cupboards there. You need which 2 sizes? A 100 and a 25. Okay? Well, no, they're smaller than 25. Okay? Everyone clear on what we're looking for? Okay.